what he did is, he took a cork, he sliced them into thin slices, and he observed under microscope. Then what he observed was a honeycomb-like structures were observed. Okay, what he named them as cellulae or compartment, which means like a room, okay, which is surrounded by the walls. Okay, and it is hollow shell. It's a hollow space. Inside there was nothing. So because he was observing a dead cell, so the cork was a dead cell. So but he gave the name as a cellulae or cellula. Later it was known as cells. Okay. But now in present context, whatever the cell we are discussing, it is a live, right? Then Antony von Leeuwenhoek, he was a Dutch tradesman and scientist. He is also known as father of microbiology. So he then, out of his interest, so he was a tradesman. Okay, he used to trade clothes. So what he observed is to observe the defects in the clothes. So he made the microscope, and out in which he was observing the defects. So during that period of time, so out of his interest, he observed some water samples and all these things, and he could find that there are small animal-like structures which are moving in that samples. So then he named them as animalcules, that means small animals. Later, the name was given as microorganisms. Okay. So he observed bacteria, protozoa, spermatozoa, red blood cells, then muscle fibers, all these protists, all these were observed by him. He actually drawn all the images. He documented all the images. Okay. Then Robert Brown, so he preserved the nucleus of the orchid cell, okay, root cell. Then slowly, this is the evolution of, like, from beginning of the identification of cell, how the cell biology was developed. Then Felix, he was a French biologist. He discovered semi-fluid substance of cell, which is called as sarcode. And then Sheldon and Swan, so they have given the cell theory. So this Sheldon, he observed that plant cells consist of cell wall, okay, and a jelly-like substance and a nucleus. Then Swan, so he discovered that animal cells lack cell wall, and both Sheldon and Swan, they contributed to the cell theory. So what is cell theory? So cell theory states that all living organisms, they start their life by a single cell. Okay. So it's made up of cells. And like each cell, actually that means a cell will give rise to another cell. That means prior to this theory, there was a notion that spontaneous generation. That means the cells evolve spontaneously, or the life evolves spontaneously, but that is not the case here. So cell theory, what it states is a cell gives rise to another cell. Okay. And each cell, it has a complete function of the organism. Okay. Then Purkinje and von Mohl, so they have renamed the sarcode as protoplasm. Proto means first, plasm means form, first form. Okay. Then the cell membrane was discovered by Schwann, and the name was given by Nagel and Kramer. Then Rudolf Virchow, he was a German doctor and biologist. He accepted the work of Robert Remarque, and he showed that origin of cells was a division because of division of pre-existing cells. As I told you, like spontaneous generation was, the theory of spontaneous generation was condemned by this cell theory. So Here, on the basis of organization of DNA, cells were of two types. One is prokaryotic cells and the eukaryotic cells. So pro means primitive, okay? Karyo means nucleus. Eukaryote means true nucleus. So what are the characteristics of the prokaryotic cell? So the DNA or the nuclear material is naked. That means in eukaryotes, there is a nuclear envelope which is surrounding the DNA. But in prokaryotes, there is no such thing and the DNA is naked and it is lying in the cytoplasm. Okay. Or nucleoid, nuclear body and genophore or prochromosome. So these are some other terms. Then additional small circular DNA entities called plasmids may be present. So usually in bacteria, the plasmids are present. And the nuclear components are completely absent. And the cell wall is present in bacteria and cyanobacteria. Okay, but not in mycoplasma. So mycoplasma, it looks similar to that of eukaryotic cell. Okay. And PPLOs means pleuropneumonia-like organisms. So the main characteristics are that the nuclear envelope is absent, DNA is lying as naked, and even the cellular organelles, they are also means highly specialized organelles were absent, and plasmids, especially in bacteria, you can also see plasmids, and cell wall is present in bacteria and cyanobacteria. So this is the prokaryotic cell, how it looks like. So what are the components? You have nucleus, that is chromosome, but it is not organized, okay? And plasmid, cytoplasm, it's a fluid-like component within the cell. Then you have plasma membrane, which is present in both eukaryotes and prokaryotes. But apart from that, it also have cell wall. Even cell wall is present in some eukaryotes like plants. Then outside the cell wall, even some bacteria, they consist of capsule or slime layer. Then some extra appendages like villi, then granules and flagellum. All these are present, which help in the acquiring food, reproduction, or movement towards some chemicals or food source. Okay, and the granules, they usually help in the storage of either like gases or food material, etc. Okay, and ribosomes for the synthesis of proteins. Yeah. So a single-stranded flagella or fimbriae is present, which is mainly for the movement. Then photosynthetic thylakoids are not organized into chloroplast, as in the case of plants. Then membrane-lined cellular organelles like mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, microtubules, microfilaments, centroles, all these are absent. And gas vacuoles, which are mainly for the diffusion of gases, is present. Then ribosomes are 70S, that is, it is made up of two subunits, 30S and 50S. 30S is a small subunit, 50S is the large subunit. Then one envelope system. Then cyclosis, that's the movement of cytoplasm is present, it's absent. Then mitotic spindles are not present during cell division. Sexual reproduction is absent, usually through fission, binary fission, or if it is through conjugation, okay? Then low DNA content. Transcription and translation occurs, both occurs in cytoplasm. Then respiratory enzymes, they are in close contact with the cell membrane. Then endocytosis and exocytosis, that means they take up something inside the cell and releasing something or secreting something outside the cell through vesicles. So that is called endocytosis and exocytosis. So endocytosis means taking inside. Exocytosis means releasing outside. So both are absent here, and nitrogen fixation is seen in some nitrogen fixing bacteria, all this. Okay. Then eukaryotic cells. So eukaryotic cells, we can see an organized nucleus, membrane covered cell organelles like mitochondria, endoplasmic reticulum, Golgi complex, all these. If it is a plant cell, we'll have chloroplast. Then cell wall is present only in the plants, fungi, and protists. Even in the protists, we don't see any muramic acid, whereas in bacteria, we see muramic acid. Then flagella, if it is present, it is 11-stranded with axoneme and sheath. And the nucleus has nuclear envelope, chromatin, nucleoli, and nucleoplasm. And the DNA is linear, which is associated with some proteins called histones, okay, which helps in the packing of the DNA. Then extranuclear DNA is, if it is present, it is usually circular. Then most of the DNA lies in the nucleus. Only small quantity of DNA is present in the mitochondria and plastids. And the transcription occurs in nucleus and translation occurs in the cytoplasm. So once the transcription is done, mRNA is synthesized, and this mRNA will travel to the cytoplasm where it helps in the synthesis of protein. Then respiratory enzymes are present in the mitochondria and cytoplasm. So this is the structure of the eukaryotic cell. So we have microfilaments, centrioles, nucleus, ribosomes. The ribosomes helps in protein synthesis. Then lysosomes, which are suicidal bags, which degrade the cells, damaged cells or components. Then Golgi apparatus. Then rough endoplasmic reticulum, which have ribosomes on their surface. Then mitochondria and smooth endoplasmic reticulum, which don't have any what you call ribosomes on their surface. So here endocytosis and exocytosis are quite common here. Then cyclosis is present, and the ribosomes are both 80S and 70S. So 80S have two subunits, smaller subunit and larger subunit. So smaller subunit is 40S and larger subunit is 60S. Whereas in 70S ribosomes, as I told you, 30S and 50S. Okay, they are also present in the plastids and mitochondria. Sap vacuoles are present in plants. Thylakoid, chloroplast, thylakoids are present in the chloroplast in the plants. When sexual reproduction is present, spindle apparatus is formed during nuclear division, and nucleus contains more than one chromosome. As you know, any organism, or even simple example is human beings. So many, we have 46 chromosomes. So these are the various components of the eukaryotic cell. Now this is again within the eukaryotes, we have plant cells and animal cells. So some components are present in the animal cells, some are present in the plant cells. I'll just read out. So in the plant cell, we have peroxisomes, then mitochondria, Golgi complex, microfilaments, then vacuoles, microtubules, chloroplast, cytoplasm, ribosomes, smooth endoplasmic reticulum, rough endoplasmic reticulum, plasma membrane, cell wall, nucleolus, and nucleus. So here, some components like chloroplast is present only in the plant. Whereas in the animal cell, we observe flagellum, nucleus, nucleolus, Golgi complex, cytoplasm, basal body, microfilament, lysosomes, okay? Then centrosome, centrioles, pericentriolar material, ribosomes, microtubules, peroxisomes, rough and smooth endoplasmic reticulum, mitochondria, and plasma membrane. So mostly they are similar, one or two components are different. So this is the here you can see that. So DNA is a genetic material and it contains ribosomes, which is common for both, and similar metabolism is seen. But the difference is prokaryotes, they consist of cell wall, single cell, and they don't have membrane bound organelles. Whereas eukaryote, they consist of nuclear envelope, cell wall, then multicellular organisms are made up of, and they have DNA. So this is the real images of what you call cells. So on the right hand side, this is a prokaryotic cell, and the left hand side is the eukaryotic cell. So it's a bacterium and green algae. So it is the same thing. I'm not going to repeat all this. So now we will discuss about various components of a cell. So first is plasma membrane. So all the cells are surrounded by the plasma membrane. So plasma membrane is also called as cell membrane, plasma lemma, or biomembrane. It is made up of partially fluid, partially solid. Okay. So here, so it is selectively permeable. That means it won't allow all the components from outside the cell, outside the environment into the cell. Okay, it is semi-permeable. It allows only some molecules, especially small molecules, ions, such kind of water, such kind of things, to some extent. Okay, that too through with the aid of various channels. Okay, that we will discuss in detail. So it is semi-permeable in nature, and it is dynamic. It is not a rigid thing. It is not like stiff. Okay, it is flexible and dynamic. It will change. The proteins will move, lipids will move. Okay, and appear trilamellar or triparticulate under electron microscope. And if it gets injured, within no time it gets repaired. And it consists of 20 to 79% of lipids, 20 to 70% of proteins, 1 to 5% of carbohydrates, and 20% of water. So it is a bilayer, 2 layers. Okay. So outside layer, which is called outer leaflet, and inside is inner leaflet. So it consists of lipids, which are phospholipids. It will have a head, 2 fatty acyl tails. So here you can see the orange ones are heads, and the black ones are 2 tails. Okay. And within these lipids, various carbohydrates, protein molecules gets attached. They are called phospholipids, lipoproteins, that means, which are associated with the lipids, proteins. Then glycolipids means the sugar molecules or carbohydrates associated with the this lipids. Okay. So they are glycolipids and lipoproteins. These proteins are phospholipids. Okay. So the lipids are phosphoglycerides or phospholipids. So the lipid molecules are amphipathic in nature. Amphipathic means, amphi means 2. Pathic means they are both hydrophilic and hydrophobic in nature. That is polar and non-polar. So hydrophilic region is the head, and the hydrophobic part consists of the 2 tails of fatty acids. Okay. And this hydrophobic tail is towards the center, forming the lipid bilayer. That means you just imagine that when the cell is round in shape, the outer surface will get exposed to the environment. Like inside our body, we have a lot of fluids. So it has to get exposed to the water. So therefore, the outer head groups are polar in nature, and they are exposed to the outer environment. And even within the cell also, we have cytoplasm. Again, that should be polar. So inside the layer also, it consists of head groups, right? So the head groups are mainly exposed to the outer environment and inner environment, and both are polar in nature. Whereas at the center of this bilayer, it is hydrophobic in nature because the lipid molecules are present. Then protein. So proteins are, here they are fibrous or globular structural, and they are carrier proteins, and they are receptors as well as enzymes. So these proteins have multiple functions. Okay, fibrous proteins or globular proteins. So globular proteins means examples like hemoglobin, myoglobin, immunoglobulin. Okay, all these are globular proteins. Then some are fibrous proteins, and they have structural role. That means in muscles, we'll have proteins which will help in the structure. And carrier means glut proteins, which will carry the glucose transporters. Then receptors. So various proteins, they act as a receptors, which are present on the external surface, which will identify some ligands or signals. And some proteins, they act as enzymes. And they also possess both polar and non-polar side chains. So polar linkages are towards the outer surface, and non-polar linkages are towards the inside, or they will be folded, right? Then carbohydrates. Carbohydrates are branched or unbranched oligosaccharides like hexose, fucose, hexamine, and sialic acid, etc. So these are the main components of the plasma membrane. So now we will discuss about the model or the theory which determines the structure of plasma membrane, which is called fluid mosaic model. So the plasma membrane consists of two layers. It's a bilayer of phospholipids. It is like a vegetable oil, so it is flexible, okay? And its temperature is also, it will change its composition based on the temperature. And it is not solid, it is quasi-fluid. Sometimes it's fluid, sometimes it's solid. And the protein molecules occur in places both inside and outside. Outside, the proteins which are attached to the outside of the plasma membrane are called extrinsic or peripheral proteins. And those which are present inside are called intrinsic or integral proteins. The protein icebergs, they appear as icebergs in the sea. That means at the tip, we can identify these proteins. And there are also these integral proteins, they form tunnel proteins or transmembrane proteins. So which will make a tunnel or a tube-like structure through which some molecules like water can be passed. And these tunnels form channels for the passage of water and water-soluble substances, and also some ions. And proteins are held by polar and non-polar side chains. So the extrinsic proteins are mostly on the cytosolic phase or on the external phase, which are called spectrin. And the extrinsic proteins are covalently attached to the phospholipid head. And if they are attached to the transmembrane protein, they are non-covalently attached. They have functional and structural specificity. And they also change their flexibility. That means they will be more flexible, and their composition also vary according to the external environment. So they are more dynamic. And the proteins have functions like enzymes, permeases, carriers, etc. And the lipids which are present are attached to these carbohydrates and proteins. Hence, they are called as glycolipids. And sometimes these carbohydrates are attached to proteins also, which are called glycoproteins. They are mainly present in the glycocalyx or the cell coat. Okay. And what is the function of these oligosaccharides? So now we understood what is the function of the lipids. Okay, it's a structural component. And what is the function of the proteins? So they have multiple functions. Then what are the functions of the oligosaccharides? Mainly as recognition centers, sites of attachment, antigens, they act as antigens, and they have negative charge so that they provide negative charge to the outer surface. So what now we come to the functions of the cell membrane. So what is the role of this cell membrane is compartmentalization. That means the external environment and cell are separated from each other through this plasma membrane or cell membrane. Then protection from injury. So directly the fluctuations in the external environment, if they are not compartmentalized, they will directly affect the inside of the cell. So therefore, to protect the cell from the injury. Then provide organic connections between the adjacent cells, like gap junction, plasmodesmata, and they are selectively permeable barrier. That is, next is transporting solutes through that. If there is no barrier, all the components will enter the cell and they will damage the cell. And responding to the external stimuli. So the receptors which are present on the cell, they usually respond to the external stimuli and they will carry out some changes within the cell. Then energy transduction. So the exchange of energy. And secretory, excretory, and waste products are thrown out of the cell through exocytosis. So these are the important functions of the cell membrane. Then membrane transport. So how these ions are transported in the membrane. So it's mainly by passive transport, active transport, and bulk transport. So passive transport is nothing but here there is no need of any energy to move particles from one side to another. It is through concentration gradient. Gradient means the change, okay, the difference. So concentration gradient means concentration difference. If the concentration of solute is more outside, so from more, that means from outside to inside, the component will move, right? So that means, suppose sodium ions are high, they will be usually high outside the cell, whereas potassium ions will be more inside the cell. So this movement, I'm just giving you the example. Actually, for sodium and potassium, we will have channels. But just for your understanding, I'm telling. So based on this concentration difference, the movement will take place. Then passive diffusion means here the cell membrane plays an important role in passive diffusion. So here, neutral solutes and lipid soluble solutes. So they can, because lipid, so the center portion of the bilayer is made up of lipid. So the lipid soluble molecules can be easily passed through the concentration gradient, that is from higher concentration to the lower concentration. And the second option is open channel transport. So here channels in form of tunnel proteins. So okay, they form tunnels, and the water channels or aquaporins, they allow this water to, and water soluble gases like carbon dioxide and oxygen to pass through the this tunnel across the cell. So through osmosis. And the filtration is through the diffusion, which is under the pressure. So here no need of any energy, but simple passage through concentration gradient can happen. Then facilitated diffusion means some agent has, yeah.
The transport is called active transport.
Passive. So active transport means, so there should be energy which is involved, like ATP. Okay, I will get to that also. So facilitated diffusion means here some agent, like if you have gated ion channels or permeases. So here some transporter will help in the movement. Okay, here energy is not required, but some agent is required. So this is also through the, along the concentration gradient. So here you can see, so the particular molecules bind to a carrier, special carriers, like for example, glucose. Glucose binds to transporter, glucose transporter, GLUT proteins. So this GLUT proteins will actually transport this glucose throughout the cell. So here this, you can see this orange color ball, okay, represented. So that will come and bind to this carrier, which will fold and which will help in the movement of this particular molecule inside the cell. Okay. So that means this is not an active transport. This is a facilitated diffusion. Not simple diffusion, but facilitated diffusion. Then we have ion channels. So ion channels are very specific. Okay, like we have sodium channels, potassium channels, which allow only that particular ions. A specific channel for each ion, they do not move in dissolved state, and most ion channels are gated. They open only in response to some stimuli. Depending on the stimuli, there are three types: voltage gated, mechanical gated, and ligand gated. So voltage gated means during action potential based on the charge. So based on the voltage, they will open. Mechanical gated means some like agent will help in the opening. And ligand gated means the ligand when comes and binds to the receptor, then it will open. So movement is again according to the concentration gradient, and the rate of passage is high across these ion channels. Then we have permeases. So permeases are nothing but the proteins which help in the facilitated pathway of movement of substances. And the rate of transport is stereospecific. Stereospecific means we have a D, L, okay, stereoisomers, right? L and D, for example, if you know the amino acids, they will have L amino acids and D amino acids. Okay, based on their configuration. So it's very stereospecific means if it is D permease means it will carry only D amino acids like that. Okay. And the saturation effect recorded. Then co-transport means two ions or two molecules are transported at the same time. Okay, like glucose and sodium. So then again you have uniport and symport and all these things. So co-transport means two molecules will transport together. So here symport means both on the same direction. Okay, uniport means only single molecule. Like that, we have again different types. Then active transport means here the energy is required in the form of ATP. So here various ions are transported then iron, chlorine, all these are transported, and the carrier proteins and the embedded proteins, they change the shape of the cell, they open and close across the cell membrane. So that means whatever the protein which is present, based on this, whenever this ATP is hydrolyzed into ADP and PI, so the energy is released. So this causes the conformational change in the protein, so that will either close or open inside that is across the cell membrane, and they will allow the molecules. Then bulk transport is in the form of endocytosis or exocytosis. So endocytosis is again of two types: pinocytosis and phagocytosis. Pino means drinking, cytosis means cell, that is cell drinking, that is fluid molecules are transported. Phagocytosis means cell eating. So the phago means eating, cytosis means cells. So endocytosis means here taking up material inside the cell. So that is taking up the other cellular components or taking up the liquid components. Then exocytosis, where release of components. It is again through pinocytosis or photocytosis. So here minute detectable vesicles with fluid matter are present, or substances, dissolved ions are present, and it's called cell drinking. And as soon as solute or ligand particles form complex with the receptors, here what happens is, you can see, I will show you. Yeah. Whenever the fluid enters the cell, so the plasma membrane that will bend inside, forming a vesicle, and the two membranes will get fused to form a vesicle, which will be released into the cytoplasm. Okay, this is how it happens. So that vesicle is called as pinosome. So this pinosome migrates inwards, and the digestion, this pinosome, it fuses with the lysosome and the digestion happens. Then phagocytosis means cell eating. So here also it forms a phagosome, and this phagosome fuses with the lysosome, and it will, the food, that is the components present in the vesicle, gets digested. So here you can see in the cell, when the microorganism or a particle comes, the plasma membrane invaginates, and it will form a fuse, like a vesicle, which is called phagosome or phagocytic vesicle. This phagocytic vesicle fuses with the lysosome, form phagolysosome. So the enzymes which are present in the lysosome, they will digest this component in the phagolysosome, and then this digested microbe and some indigestible material will be present in the residual body, which will be released outside the cell, and all the like the material which is not digested, that will be released outside the cell. So this is nothing but the neutralization of the microorganism by immune cells. Then exocytosis is nothing but exportation of the substance from inside to outside. So here you can see the secretory vesicle from the Golgi apparatus consists of some material which will fuse with the plasma membrane, and they will secrete that material outside the cell. This is the exocytosis. Here there are mainly two important cells, nerve cells for neurotransmission and secretory cells, especially in the insulin, Langerhans, beta Langerhans. So this is how the exocytosis takes place. So this is all about the plasma membrane. Now, when comes inside, so inside it is the cytoplasm. So this cytoplasm, or it's also called as cytoplasmic matrix or cytosol or hyaloplasm, so which consists of a fluid, clear fluid, and the functions of the cytoplasm are mainly to provide raw material to cell organelles and exchange of materials between the cell organelles, then biosynthesis of fats, nucleotides, carbohydrates, proteins, and coenzymes, then catabolic activities like in some primitive organisms, we can see glycolysis, anaerobic respiration, PPP, that is pentose phosphate pathway, and other respiration, all this occurs in the cytosol, and distribution of various materials inside the cell via cytoplasmic streaming. So these are the various functions of the cytoplasm. So what are the cellular organelles present? So these are endomembrane system. That means within the cell, again, membrane-bound organelles are present, especially endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. So the endoplasmic reticulum was discovered by Porter and Thomson. So it's a 3D structure. Okay, it's interconnected structure which is present throughout the cytoplasm and consists of cisternae. Okay, so here there are two types: smooth endoplasmic reticulum and rough endoplasmic reticulum. So the smooth endoplasmic reticulum consists of no ribosomes. Okay, also called as agranular endoplasmic reticulum, and rough endoplasmic reticulum consists of ribosomes, which are called granular endoplasmic reticulum. So it consists of cisternae, which are flat interconnected sac-like structures, and then vesicles, which are oval and rounded sacs, which are also called as microsomes, and tubules. Okay, tubules are nothing but tube-like extensions connected in the cisternae or vesicles. So here is the figure. You can see there are cisternal space. So these foldings are called cisternae. So whenever the ribosomes are present, they are called rough endoplasmic reticulum, and where these ribosomes are absent, that is called a smooth endoplasmic reticulum. So the functions of endoplasmic reticulum are They provide large surface area for various physiological activities. They provide mechanical support to the cytoplasmic matrix, and they provide quick intracellular transport, and they are the provide precursors, that means starting material for secretory substances to Golgi apparatus, and they give rise to vacuoles. And this rough endoplasmic reticulum, they actually synthesize and form glycoproteins, where the sugar moieties are attached to the proteins. And smooth endoplasmic reticulum are sites of the synthesis of fatty acids, phospholipids, and steroids. And it is also called a sarcoplasmic reticulum, and which stores calcium during muscle contraction. And these endoplasmic reticulum helps in the detoxification of various chemicals. Then Golgi apparatus or Golgi complex, which is made up of smooth membrane like sacs and cisternae are present. And it was first seen by the George, but named after Camillo Golgi, okay, who actually recognized reticular structure in 1898. So the functions of this Golgi apparatus are all granular cells depends upon Golgi apparatus. That is the secretion, the package of the proteins is mainly done in the Golgi apparatus. Formation of glycoproteins, glycolipids, and formation of sialic acid, galactose, fat transport, then mediation of hormones which are produced by endocrine glands, then lysosomes and secretory vesicle formation. All these things happen in the Golgi complex. So this is how the Golgi complex looks like. And here you have the cis phase and the trans phase. Okay. So the incoming transport vesicle enters through cis phase, and the vesicle with the packed protein is it exits from the trans phase. Okay, so the secretory vesicle, it exists from the trans phase. Okay, so here you can see how it happens. So here the vesicle is transported across the Golgi, and so this secretory vesicle, it comes and fuses with the plasma membrane, and exocytosis takes place. Okay. And even the lysosome, which is having the material, the endosome. So through endocytosis, whatever the vesicle which enters the cell, that fuses with this vesicle of the Golgi, and it will form it further fuses with the lysosome, and the digestion takes place. So the lysosomes are single membrane containing hydrolytic enzymes containing organelle. So they are called suicidal bags, which have acidic in nature, that is pH will be usually 5. It is formed by the Golgi complex. So here mainly the digestive enzymes are present in these lysosomes. So the functions of lysosomes are intracellular digestion, that is food obtained via phagocytosis, and then the extracellular digestion by release of enzymes via exocytosis, then defense. So the lysosomes of leukocytes, white blood cells, they usually excuse me, protect the cells host from the foreign material by the digestion of foreign proteins, bacteria, and other toxic substances. So here we have autophagy, autolysis. So autophagy is nothing but it is like metamorphosis of many animals. So where the embryonic parts are digested. So even for the human beings also, or any other organism, first a ball-like structure will be formed. So that ball-like structure has to form into a particular shape of the animal. So during that point of time, the extra cells has to be digested. So that is done by these lysosomes. Then autolysis. So it's formation of thyroxine active hormone. So thyroxine, which is active hormone of thyroid. Okay. This is mainly through the hydrolysis of thyroglobulin. So thyroglobulin is a proenzyme, okay, or prohormone. So that means it is inactive form. So that has to be converted into the thyroxine by this action of the lysosome. Then vacuoles. So vacuoles are non-cytoplasmic areas. So it has sap vacuoles, different types of vacuoles, contractive vacuoles, food vacuoles, air vacuoles. So sap vacuoles are nothing but the fluid-filled vesicles separated by tonoplasts. Then contractive vacuoles, which occur in algal cells and protists. Then food vacuoles are present inside the prokaryotes, which fuses with the phagosomes and lysosomes. Then air vacuoles, they are pseudo vacuoles and mainly for the gaseous exchange. Okay, I'll stop here. So if you have any doubts, you can ask.
Ma'am, once again, you explain like types of lysosomes.
Types of lysosomes.
Is there in page number 22 in the reader textbook.
Sorry, I don't have textbook right now. So
shall I read? Will you explain?
yeah, yeah.
Shall I read, ma'am?
Yeah, you can.
Okay. So the first one is primary lysosomes or storage granule is a small newly formed organelle from Golgi, contains enzymes synthesized by lysosomes and accumulate in the endoplasmic reticulum. From here, they penetrate into Golgi complex.
Okay.
Then secondary lysosomes, also known as heterophagosomes or digestive vacuoles, or are produced either from phagocytosis or pinocytosis of foreign material by a cell forming phagosomes or pinosomes.
Okay. Only two?
No, ma'am. C is also there.
Okay.
Autophagic. This you have explained, ma'am. The autophagy.
Okay. Yeah, okay. Only
Then,
three, no?
no, ma'am. One more is there. D. Residual bodies are formed by incomplete digestion of foreign substances and are huge, irregularly shaped and electron dense. In some cells, defection or exocytosis eliminates the residual bodies.
Okay. So I think I also explained about residual body somewhere. Okay, anyway, I will explain. So here, primary lysosomes means, as I told you here, they are formed mainly from the Golgi complex.
Okay.
So I will just a second. Yeah, here you can see in this image. So here, through exocytosis, whatever the material enters, so that will again, this endosome, they form a vesicle which is called endosome, which fuses with the Golgi. And now, when the fusion takes place, so that will form the lysosome. Okay, this is the primary lysosomes. So secondary lysosomes means phagocytosis or pinocytosis. That means especially in the immune system, our immune cells, whenever a bacteria or virus, something enters inside our body, so they will, there are some phagocytic cells within our body, inside like immune system. So like dendritic cells, macrophages, all these things. So they will actually, phagocytosis means cell eating. So they will eat the bacteria. Okay, they will make it into a like vesicle. That phagosome then fuses with this lysosome. These the enzymes which are present in the lysosome, they will digest this bacteria. So I think I have explained this. Just a second. Yeah, here you can see. So here, whenever a bacteria or something enters, this forms a phagosome, and this phagosome, it fuses with the lysosome, and this lysosome it consists of some digestive enzymes. So these digestive enzymes, they will digest this bacteria or any microorganism, and some material will be undigested. So now, as per the text you have read, so this phagolysosome, they will be the secondary lysosomes. Okay. And third is, as I told you, autophagy. That means during our development of an organism, the lysosome will help in eating the self cells. Okay. And the fourth one is residual body. So the it is also a vesicle with undigested organism, undigested material. Okay, which will be released outside the cell. Is it clear?
Yes, ma'am. Thank you, ma'am.
Okay, okay. Okay, we'll continue tomorrow. Okay.
Yes, ma'am.
Yeah,
Structure and function of cell
yeah,
organelles. We are having.
yeah. We have many more organelles, but as time doesn't permit, so we'll continue tomorrow.
Okay,
Okay.
ma'am. Thank
Yeah.
you, ma'am.
Thank you. Thank you.